Okay, our last speaker is our keynote speaker. It's Willard, who is no stranger to Stanford. He's been a bit of a journeyman, so to speak, over the years. I think Toronto, Stanford, Case Western, Duke. Now he's actually got a very interesting position he's taken recently at Geisinger. So he's a pioneer, as I think all of you know, in the EXIST field, having helped discover this and made many seminal findings. So I guess he's going to give us the latest and greatest today, sex chromosome view on human biology and medicine. Hunt.
Do we care which of these we use here? No, I move.
He'll turn it.
Okay, is that working? Apparently. Thank you, Mike. Thank you, Marcia. Great to be back on this campus, although every time I come here it looks like nothing I remember from back when I used to be here. And this has been a great day of talks, and I hope not to ruin it now in the next 30 or so minutes. I took as my charge to sort of give an overview of much of what you have heard, given that I'm coming at the 3' end of the day instead of the 5' end of the day, but to provide a context from someone who is, I confess, a human geneticist from the beginning, trying to explore human biology and medicine and using human systems and human patient material, which explains this most recent move of mine. And I'll sort of take us through that as we go. I'll move quickly through some areas of my talk only because it's been covered by some of the others. And I start with this. I was convinced when I started as a human geneticist, and that started when I was an undergraduate 100 or so years ago, and my first project was on exon activation. I don't think I even knew there were autosomes back then. I just figured this is the way it was, meant to be. But came to learn during graduate school and after that most of the field of human genetics and medical genetics saw the world through an autosomal lens. That the biology and the nuances of genetics for the sex chromosomes and what we now know are differences in biology due to the sex chromosomes were completely glossed over. And incredibly intelligent and accomplished professors would stumble over the fact that, oh yeah, I forgot that it's different and you only have one X chromosome, or I forgot that there's a Y chromosome. I was quite stunned by that and figured I had a career or so to try to contribute something to that. So my interest in the particular questions came up when I joined what was then the Institute of Medicine committee, now the National Academy of Medicine, to explore sex differences. At a time in which there was a great gnashing of teeth, I was actually, it turned out, they didn't tell me at the time when they called and said, would you join this committee? I said, of course. They said, we need a geneticist. I said, well, of course. And they only later did I find out I was the third choice, because the first two geneticists who had joined had then quit because of the difficulties of the discussion between the taste great and less filling side of the debate on where on this spectrum between genetic influences and social influences lay the very obvious differences that anyone sees if you've ever met both a male and a female and you recognize that there are differences. I'm a geneticist. Like a man with a hammer, everything looks like a nail to me, and so that's my open disclosure that I start at this end, and that's the role I played on that committee. I learned a lot. It gives you an enormous degree of respect for interdisciplinary science and interdisciplinary discussion and learning to trust people who have learned other information that they're bringing to the topic at hand, and very much that's the case here. And so I've spent a lot of time at this end experimentally, but at this end in terms of trying to interpret exactly what those data might actually be portending. And it's endlessly fascinating if you're a human studying humans, as I am. So I want to cover two areas of territory. One is this concept that we've already touched on earlier of exon activation and the impact that it may have on sexually dimorphic genes and the possible effects that those genes would have, a quote, problem, unquote, that we don't see for the vast majority of genes on the boarding autosomes. And then lastly, turn to approaches for how to explore a genomic contribution of such genes to explore sex differences in biology and medicine. And this is something which, as a human geneticist, has always been exceedingly difficult to do, and in the realm of very descriptive studies. What we heard from Art, for example, where you can actually design your experiment, test your hypotheses, and make very significant conclusions coming from those experiments that you have planned. I think we're only now getting, in the age of genomics, we're getting to that stage in human genetics where we can test tens or hundreds of thousands of individuals and collect the kinds of information that we would need in order to truly test hypotheses. So this has already been covered largely by Melissa and her terrific talk on the evolution of sexually dimorphic genomes, where it came from, this need for, or an obvious benefit evolutionarily, of dosage compensation for at least some genes on the sex chromosomes. It's not all, as other speakers have alluded to. I'm always asked, well, how many genes does it take? Why is there dosage compensation if you know you don't need to compensate for all of the genes, and presumably you don't just need to compensate for one gene, or evolution would have come up with much simpler analyses? How many genes is it, and what are they? Well, that's what I like to describe as job security, because that's a question that is going to take eons to address in a system in which we can construct the sex chromosomes the way we would like them to be, move genes around and actually test but whether they can be subject to an activation or not, once we understand the actual differences on what controls that, to be able to go through that analysis for each and every gene. There's only 1,100 of them. There's an infinite number of graduate students and undergraduates. This is simply a matter of time before this experiment is done in some system that would allow us to address exactly how many there might be. What I find fascinating is, in fact, that there are at least three, there's more than this, but the three I've got here, three evolutionarily distinct approaches to dosage compensation. So this, more than any other argument, argues how incredibly important this is to survival of many species. The fundamental mechanism in fruit flies of upregulating expression from the single X chromosome, to worms where you're downregulating expression from the 2X chromosomes, so they're only cranking out half the transcripts each in order to equalize dosage at this end. And then in mammals, something fundamentally different of randomly picking, as we've heard already today, of one or the other of the X chromosomes, so that the sum total of this would more or less approximate the single X chromosome found in males, as Mary Lyon appreciated. And life, when Mary, this is true of all hypotheses, of course, although I'm amazed at how many people, I went to a talk once on sex determination decades ago. And this was just when SRY was coming along. And we had listened to all these talks of sex determination in a whole bunch of organisms in which, in Drosophila and in worms, and you've got all of these genes, they've been identified, you've got these very incredibly complicated pathways understood in excruciating detail, but fascinating detail. And then David Page stood up to talk about sex determination in mammals and in humans. And it was a very simple one that sort of said, female, add SRY, turns into a male. And so one of the questions came up at the end and said, why is it so much simpler in mammals? You might think mammals would even be more complicated than all these other sort of little organisms. And there was this sort of dead silence for a long while, probably David trying to think, how can I answer this without insulting the person asking the question? That the simplicity of the slide was simply a function of how little we understood. And when you don't understand anything, you go from point A to point B, you draw an arrow between it, and you put your favorite model in the middle. Once you do understand it, of course, life gets infinitely more complex. And all of these stories have become infinitely more complex, but all to the good as we've understood, the field has understood different parts of it. So this is an ancient slide of mine, but this is suitable. I use this for medical students. It works for them. It'll work today. On the human X, about 1,100, 1,200 genes and 155 or 60 megabases of DNA. It's a phenomenal example, and this is what drew me to the field initially when I was an undergraduate and was just reading the literature and came across things. And who knew that genes could be regulated on a chromosomal basis, as opposed to in the early era of molecular biology, it was all about single gene regulation. What was happening at the promoter, what was happening at the enhancers, what would turn a gene on or off. It was all about one gene, one gene, one gene. And Bruce Cattenack, a wonderful geneticist in the UK, was among the very first to begin to articulate that there was a whole nother level of gene regulation, which was not at the level of single genes, but at the level of the chromosome as a whole, and the genes just sort of went along for the ride. I was fascinated by that as a student, and I guess I'm still fascinated by that as a student, and so that's the field that I ended up going into. So we know that exon activation in mammals requires the presence of what's called the exon activation center, which is located about the midpoint of on the human X chromosome, and as Barbara already pointed out, with the exist gene that we isolated by a positional cloning approach, because we knew from studying human patients, and only human patients, that it had to lie in this part of the chromosome, and we went through looking for a gene without any hypothesis of what that gene might be. How would we know it when we saw it? And I pronounced at a lab meeting with great authority that we'll know it when we see it because it'll have to show some feature that just looks different than what anyone has seen before. Oh, I had no idea what I was talking about, but it sounded really good at the time, and convinced the group of graduate students to do this, most notably Carolyn Brown, who was the single person most responsible as a graduate student at that time for identifying and convincing us about the exist gene. That was about the time I moved here to Stanford with Carolyn, because she was too early in a really important and interesting story to just abandon ship after finishing her graduate work. And we got to the point of saying we could not find, this is a 18,000 base pair transcript, base transcript, sorry. And we were sequencing through this. This is the old-fashioned way of sequencing, none of these, whirligig machines that you'd have done by Tuesday afternoon. This was old-fashioned sequencing with a lot of people who then took up having to put on glasses as they had to read those sequencing gels. And they could not find an open reading frame. There was no open reading frame at that point, we were about halfway through the transcript. And the question was, do we dare propose the possibility that this might be a non-coding gene? Now, there were no other non-coding genes. I mean, there were ribosomal RNAs, but they're the exception to every textbook that's ever been written. And so did we dare think this was true? And it was actually conversations I had here shortly after we moved here, talking to David Botstein and a number of other people who are much smarter than I was or am, citing all of these examples in other fields that made it okay to actually hypothesize that there might actually be functional RNAs, so genes whose product was an RNA, not a protein, and it would have an effect that way. And that was the beginning of the exist story. And later demonstrating that some of the genes, as we've already talked about, escaped exon activation. The key finding, and I'm going through this in the humans because, again, I think this is now, we're seeing this era where human genetics is coming back into vogue, not because we don't need mouse genetics, not because we don't need fruit flies and worms and pick your other favorite organism, but because we actually can study the human to learn about humans. And we can design experiments that are actually much more compelling than they ever could have been a while back. So I've already described the exon activation center and what might be going on here. With spreading of the exist RNA, the RNA itself stays in the nucleus. It coats, whatever coats means at a molecular level, up and down the length of the chromosome, and leads to the three-dimensional conformational changes on the inactive X. That RNA never gets to the X chromosome, the other X chromosome, even if it's the identical X chromosome, in the case of someone who's truly homozygous for the two, in the same nucleus. And of those three examples of dosage compensation in the worms, the flies, and mammals, it's the mammal that's the only example where a distinction has to be made between the two X chromosomes in the same nucleus. In both fruit flies and in worms, there's a chromosome-wide change, but it affects both of the X chromosomes. Well, a single X chromosome flies, gets upregulated, but in the worms, the two X chromosomes, and they both get downregulated. There's no distinction between one and the other. So it's actually, from an evolutionary standpoint, to be discussed only over beer or a glass of wine at this point. So why, I mean, this is a much more complicated system involving what we now know. There's thousands of examples of these long non-coding RNAs, some of which have been demonstrated to have functions, many others of which may not have any functions, or we'll just figure those out later. But this is, it just seems like a much more complicated system of having to choose between the two X chromosomes. So there has to be an evolutionary benefit. So once evolution had, quote, decided that inactivation of a chromosome would be the way the dosage compensation would be carried out, then it seems that there's an obvious benefit if you do a random choice, because that balances out the possible deleterious effects of inactivating one and only one in all of the cells, which is more what we see in the marsupial system, which is, granted they can hop, but other than that, what can they do compared to what we can do? So there's obvious advantages to having a random inactivation system, and you get, in theory, monoallelic gene expression from that X, from the active X chromosome. There are rare patients that are described in the literature that pop up every once in a while, where a female has an abnormal X chromosome in addition to its normal X chromosome, and the abnormal X chromosome is missing the X inactivation center, and therefore should not be able to inactivate that chromosome or any of the genes on that chromosome. So what happens, as you'd predict by that sort of deletion genetic experiment, you get no X inactivation, and you end up with biallelic expression from the genes that are present in two copies. And these are always deleterious. It is with a very high degree of clinical anomalies and defects in those young girls. They're almost all young girls because they don't survive into adulthood. And it tends to be relatively small parts of the X chromosome that allow, are consistent with survival with biallelic gene expression. So, as we've heard, not all genes on the X undergo X inactivation, and what determines which ones, and if it's the individual gene, or what regions of the chromosome, if it's more of a sub-regional decision, what determines whether they're subject to inactivation or escapes inactivation. There are any number of examples. They've been worked out for individual genes, but I'm not aware that it's been worked out as a sort of all-encompassing hypothesis to really explain what the epigenetic rules are of the game for the chromosome. And if you just took a gene from this part of the chromosome and moved it down here and somehow could put it over here in a very different genomic landscape in the chromosome, what's the dominant effect? Is it that region of the chromosome, or is it the particular very short-range effects of that particular gene that matter? And it may be that every gene will have its own answer. There's absolutely, as attractive as all-encompassing hypotheses are, there's actually no guarantee anywhere in your textbooks or our license to be scientists that suggests that simple hypothesis. In fact, most of the time, we learn that's not going to be true. So these studies, so Laura Carroll was a Stanford graduate student, joined my lab shortly after I got here, and she set out to develop systems to test in human material whether genes were subject to inactivation or escaped inactivation, because we knew at that time there were a few examples that escaped inactivation. And her very real contributions to the field were the demonstration of two things. One, the number of genes, the proportion of genes, she estimated about 15%. Now, with a paper that was alluded to earlier that was published last year from the GTEx consortium, now it's probably closer to 20 to 25% of genes that escape inactivation. This is where graduate students earn their stripes, right? So you take a textbook and you take the literature, and it always tells you that it's a rare exception that genes escape inactivation. And then you do the experiments and you go, oh crap, it's wrong, I got the wrong answer. It's many more genes that escape inactivation. And it turns out, in fact, to be the right answer, not the wrong answer. So up to a quarter of the genes escape inactivation. And she also demonstrated that there were a number of genes that had variable inactivation, both within a female, for those females for whom we had multiple tissues, and between females. So that you had, it was a polymorphic trait, if you will, of a gene that would have escape an activation in some individuals in the population and be subject to an activation in other females in the population. Whether that's under genetic control or not, which of course as a geneticist I firmly believe as an hypothesis would be way cool. Whether that's true or not isn't known. It could be due to something else, but it breeds true, if you will, in families. I mean, they run like segregated traits, but it could be an epigenetic trait rather than a genetic trait, given the small families that we have in our species. So that, to my knowledge, it remains to be determined. And we now know that from this paper, a dozen years later from Laura's work when she was here, that this variability is much more dramatic at the tissue level than what she demonstrated between different people. So here's all the data you ever wanted from the GTEC paper published last year. And I should say I wasn't involved in that consortium. I was only involved with semi-panicky phone calls and emails trying to explain some of the data they were coming across. Either they wanted reassurance that it mapped onto what we thought was happening in exon activation, or that in fact it sort of rang true with me. I suppose they also didn't want me to review the paper, and it worked, so it was okay. So here's from the tip of the short arm down to the tip of the long arm, the most salient features of this graph, because you're not going to be able to see it other than the fact that it's colorful. This is the pseudoautosomal region, which shows significant male bias in expression. And this is the rest of the chromosome. And when I started out, according to the rules according to Mary Lyon, the rules according to textbooks, the rest of this slide should be white. It shouldn't be colored at all. So the fact that there are any colors here tells you that all kinds of interesting things are going on in terms of expression, where many genes are upregulated in the female, overexpressed in the female with the 2 X's. Those are the ones we would conclude, quote, escape and activation. But there are others that are more highly expressed in the male with the single X chromosome. Some of these are XY, have XY homologs, but most of them do not. So there's actually all kinds of variation going on here. This is the easier to read version of the data. So enormous variability among tissues and between individuals. And the most important take-home lesson for this symposium is, I think, whatever causes that is a part of human biology that is only accessible to females. Males don't have exon activation, and so they miss out on the ability to have tissue variation and inter-individual variation for at least the genes that are on the X chromosome. I find that endlessly fascinating and significant. That's half of the species who just get some part of gene regulatory control that the other side doesn't get at all. Feel gypped. No. So the basis for that remains to be determined, but it's actually quite stunning to me. The second point here is another important take-home lesson. Dosage compensation, despite what textbooks, including my own, have been saying for a long, long time, is substantially incomplete. It isn't. There isn't real dosage. It's an attempt, perhaps, at dosage compensation. It's an accommodation to get towards dosage compensation for genes for whom dosage sensitivity is a very real thing. But both across the X chromosome and at the level of individual genes, it's quite incomplete. And so there are significant expression differences between the sexes, both for sex-linked genes and some autosomal genes, which I'll come to in a moment. And so notwithstanding exon activation, notwithstanding dosage compensation, there's a lot of sex chromosome biology, therefore, that may contribute, little evidence yet that it does, but may contribute or may underlie the evident sex differences we see in health and biology, and I'll pivot now to that part. So I come back here, and again, just remind everyone that I'm stuck way down here at this end of this spectrum, and welcome lots of discussion, as I did 20 years ago, for what happens along the full length of this spectrum. And I was somewhat pilloried in a book that was published, and Twitter screaming and yelling after that. So Laura Carroll and I constantly would refer to this as male and females. We're obviously the same species, but we effectively have different genomes. And we have different genomes. You might say it's the minority of genes, but it's fundamentally different genomes, both in terms of expression, where males have, let's say, 1,200 X-linked genes in one dose. Females only have 900 of those in one dose, maybe 300 of those in 2 doses. We've each got 25 to 50,000 autosomal genes, depending on what qualifies as a gene. And then males get this little grab bag of 50 Y-linked genes, a few dozen of those, which are not essential for life, obviously, because you females do very well without those, but that nonetheless contribute a lot to the underlying biology. So our genomes are obviously very similar, but not identical. So the geneticist view of that, there are a number of different mechanisms, and any or all of these could apply, but there are fundamental distinctions between those of us who have an XY composition and those who have an XX. So one, the presence or absence of a paternally inherited X chromosome. The presence or absence of the Y chromosome, dosage and expression of X-linked genes that we've discussed, mosaicism due to the random inactivation in females and not in males, and then sex-biased gene expression around the genome and in many, many tissues. So this is not a germline thing. This is all the tissues in the body. So fundamental differences with possible mechanisms that could play a role in many of these. And this sort of provides a mechanistic framework, a genomic mechanistic framework for thinking about what might be going on. These are more on the genetic mechanisms, and these have been demonstrated to different extents for different genes, but the relative contribution, I mean, it really would require a full-bore account across the entire chromosome to really understand how this works in the human, and I think you'd have to do the same thing. Art alluded to some of this in his talk, because he and I have been thinking about these same questions for a long, long time. And we need answers in mice just as we need answers in humans. But I always am forced myself to remind people there are 150 million years of evolution between that little mouse and a big human, right? So the last common ancestor 75 or 80 million years ago, but you've got 2 lines of evolution. So 150 million years of evolution. So there's no reason to think that you should get the same answer in mice and humans. Mice are incredibly important for shaping questions and providing mechanisms to think about for the human, but that we still have to go figure a way to answer them in the human if we want to understand the human. So now I think there are changes now that we have the human genome sequence, now that the technology of sequencing is so vastly improved and dropped in cost. There's actually an opportunity for large-scale studies of gene expression and clinical phenotypes in males and females. The GTEC consortium took care of the front half of this, but it's only now that we can begin to look at clinical phenotypes in a large enough number of males and females in order to look for either genome-wide or sex chromosome-specific effects. There's a growing number of biobanks with genomic data from just huge numbers of many hundreds of thousands of patients. The impact that the UK Biobank has had on the life sciences literature in a relatively short span has just been mind-boggling to watch. It says great things about scientists when they are open with their data and are willing to share it instead of hoarding those data, and hopefully other groups will learn from that. Decode in Iceland has been another one of these projects which has been incredibly productive. But the way that it was designed by the government in Iceland makes it very hard for others to gain access. So I do not point fingers at the scientists at all. This was just the way it was set up. I don't think the question was really thought through deeply, except in convincing the public in Iceland that they should be willing to do this. Part of this was there were privacy concerns and things of that nature, and that the benefits should accrue to the Iceland population first and foremost. That's an understandable policy statement, but it's obviously different than what is happening in the UK, and different from what we're trying to do now that I've switched my own position to Geisinger, a health system in the middle of Pennsylvania, which started 10, 11 years ago to create a biobank, which now has 200,000 of their patients who are consented. 95,000 of them have had their full exomes sequenced in a collaboration with Regeneron, who has a spectacular laboratory. And the hope is to crank through up to between 250 and 500,000 of the Geisinger patients. The position I've taken in the Washington, D.C. area, something that we call Geisinger National, is to work with the system at the hospital, and no doubt with lawyers, to figure out how we can take our database and make it open. Regeneron, and absolutely no criticism, because their mission is to have access to that information. They paid tens of millions of dollars for all the sequencing that's been done, and will pay more for more of the sequencing. And so naturally, they want the sort of first dibs to be able to recoup their investment. All fair. But that's a very different mindset than we have, where we want to be able to take what we hope someday in time will be millions of patients of sequence data and health data, where at Geisinger we have a 20-year-old electronic health record. So the average patient at Geisinger has 14 years of electronic health data in the EHR. That's an extraordinary record of longitudinal health care for taking any question you want and exploring it. And what I complained about for years, and then they finally said, well, if you think you're so bloody smart, take the job and you figure out how to do it. What I complained about for years was they could hire every smart person they could find, and they'll never answer all the questions. There's so many questions that come out of that kind of a data set that the only responsible approach is just to open it up to everyone and let every qualified investigator follow whatever their particular question du jour is, and use the data and figure out what they want. And they can toggle back and forth between the UK Biobank and the Geisinger system. They can get replication studies done overnight, and see where that takes us. I think that would be a phenomenal step forward, and we just have to figure out the mechanisms for doing that with the sequencing data. The EHR data we could do tomorrow. The sequencing data will lag behind that, and we're going to have to figure out that. But that's what I'm trying to do up there. But because of the numbers, that if you focus here, which is where, as some of you know, a lot of my initial interest in human genetics came from, with sex chromosome aneuploidies, which are very common genetic disorders, 1 in 500 overall, right? All the diseases that people at Stanford and elsewhere study, genetic diseases, it's a tiny minority of them that are anywhere near that frequent, right? We study ones that are found at 1 in 10,000, 1 in 20,000, and we call them common genetic diseases sometimes. So this is the common one, and in fact, I think it allows in the human to do what Art described so eloquently in the mouse, for exploring a chromosome effect versus the sex effect. And I'll take my last moments just to touch base on this. I'll pass over this quickly. This is another one of the GTEC consortium papers, simply demonstrating the amount of sex-biased gene expression. The controls here are the Y chromosome, which are tilted way to the male side, and the exist gene tilted way to the female side, so those are your controls. But look at all the other genes that have sex-biased gene expression. And the blue ones are autosomes, autosomal genes. So there's a non-trivial number of genes all around the genome that have sex differences in their expression. And obviously, everyone would love to know the difference between those. This one I'll skip over. We know that there is a lot of sexual dimorphism at the phenotypic level. Some of these have already been touched on by other people. Some are present more in males than in females, some in more females than males. And the actual basis for any of them in terms of the sexual dimorphism, the mechanism underlying that sexual dimorphism, is only understood in very few cases. But let me come back to this, because now that we can explore individuals who carry an unusual number of X's and or Y chromosomes in numbers. So in the Geisinger biobank, for example, we have hundreds of cases that are sitting there who have a diagnosis, not a clinical diagnosis, but a genotypic diagnosis for whether they have an imbalance of X or Y chromosomes. So it avoids that bias of saying, well, this person clearly has Turner syndrome because we diagnose them as having Turner syndrome. This way we can do that at the genomic end and drive the questions that way. So XY males, XX females. There are a small number of individuals, this was mentioned previously, about 1 in 10,000, 1 in 20,000, who are XY females, not XY males. And similarly, with the same frequency, there are XX males instead of XX females. So this comparison allows one to explore the effects of sex itself, male versus female. The genomes are nicely controlled here. It's only the sex that's different between them. Here you can look at the same sex, male, but different sex chromosome composition in order to look at some of the effects there. And similarly, X versus Y on this side of the equation. Those are rare, 1 in 20,000. You're going to need a lot bigger biobank in order to get reasonable numbers there. I'm much more excited about these ones here with Klinefelter syndrome, XXY males, XYY males here. I'm a little less excited about Turner syndrome for a number of reasons. They are less common, high frequency of mosaicism, and a lot of differences in different tissues for mosaicism, and now I've got to worry about the placenta as well. So this is getting, it's much more complicated, this system, than Klinefelter's and XYY syndrome. XYY syndrome, a frequency of 1 in 1,000 male births. That's quite common. That's 3 times more common than cystic fibrosis, for example. In Klinefelter's, a frequency of about 1 in 600 male births. So a large number of patients that are available, regardless of whether they have a phenotypic presentation or simply a genomic one, as is shown here. So here we can look directly at the effect of the extra Y chromosome. What impact does it have? And here the effect of adding an extra X chromosome, and here the effect of adding an extra Y chromosome as well, but now against an XX background instead of an XY background. So it sets up for a reasonably well-controlled series of experiments for comparing the underlying biology in those different groups of individuals, armed with phenotypic information from electronic health records and genotypic information from the whole exome sequencing that we have already done on these. And so this just shows you, this is from an earlier time when we had maybe 45 or 50,000 whole exomes done. So this is karyotyping by DNA sequencing, not by chromosome analysis. You've got a large number of females here who have, this is on log scale, so an intensity here for the X chromosome and essentially no Y chromosome. The other big bolus here for males who have a Y chromosome, but a smaller peak for the X chromosome. Here are your Klinefelters that pop up, easily found. Some of the Turner syndrome ones easily found, and some of the XYYs that are out here easily found. So I don't think it's going to be hard to find them here. Same data essentially from the UK Biobank so far. So I'm particularly interested in focusing on Klinefelter syndrome because a simple question. Do they show the male or the female incidence of sexually dimorphic conditions? It should, if in a simple world, they should be one or the other. Or they could be in the middle. That just makes it more complicated, but rather than just the male or the female incidence. The most robust examples are osteoporosis, lupus, Sjögren's syndrome, who have 5 to 10-fold differences in the incidence between males and females. The molecular, this is sort of the proof in the pudding, a wonderful paper published last month and demonstrating the molecular basis of lupus in these and the effect of an X-linked toll receptor gene, which where it had already been demonstrated in model organisms that the overexpression of the toll receptor gene would sensitize mice to lupus. They showed that the gene escapes inactivation in B cells and could demonstrate by other means of overexpressing the gene or underexpressing the genes would toggle between lupus or no lupus. So at least for that one example, very clear data implicating exon activation and an X-linked gene underlying the sex difference in the frequency of this particular disorder between males and females. Whether that will be nearly as clean for a whole bunch of other genes and conditions, we'll have to find out. So the chromosomal basis for sex differences that we've talked about today, I think it's an increasingly, certainly compared to when 15 years ago when I was on that IOM committee, an increasingly well-known and appreciated set of differences between males and females. And there's now clear, robust evidence for sex differences in gene expression and the kind of role that I just outlined of exon activation and X and Y linked genes. But in most cases, the biological mechanisms and the responsible genes and pathways, is it all genes on the X and the Y? Is it just 6? Is it, what are they? What are the key players and how can we understand what their mechanisms are and exactly how they explain those sex differences that we see? So thinking and talking about and thinking of designing experiments to explore sex differences, I think, is no longer completely taboo in the sort of post-Larry Summers era. That's an East Coast joke, but sorry. And I think but to me, as someone who's been thinking about these questions, not articulated today the way I would have articulated them when I came into human genetics or when I was here as a faculty member at Stanford, I mean, this is an existential human question, right? I don't think that people would worry quite as much about it in male and female Drosophila or even male and female mice. It really is an existential human question. For half of our species that is different from the other half of the species, and what, we somehow embrace that both scientifically and otherwise and determine exactly how that plays out. I think this calls now for really rigorous, in-depth, unbiased studies, and I think the good news is we can do those human studies now to explore the biology in a way that couldn't have been done even 5 or 10 years ago, much less 20 or 30 years ago. But recognizing that kind of interdisciplinary research will engage both scientists and society, and should, and it will be exactly that sort of sweet spot of where science and society come together and should come together in order to better understand our species and how we function together. I think, nonetheless, it's still a largely untapped area of human biology and medicine, so this is, in fact, job security for students. I think it's endlessly fascinating. It's an existential human question, and you've got a whole career ahead of you to figure out how it works. And my bet would be, I won't be around to watch it, but my bet would be we won't understand it a generation from now. This is going to be one of these gifts that keeps on giving as we try to sort it out. And it's a terrific example of what drew me into human genetics initially, of clinical phenotypes and the kinds of genomic and genetic variation that provide a spectacular opportunity of nature to provide those variants that will inform biology and inform our understanding of ourselves. And that's, we are the only species on Earth that gets to do that kind of stuff, and I find that sort of self-affirming for a question that is so important as this particular one happens to be. So I'm back where I start. What used to be a genetic framework is now very much a genomic framework from my perspective, and I look forward to the next decade or so of research that goes on to this field. And so thank you for having me here.